This is the second of two papers researching the sublethal effects of oil spills. In this review, we will be looking at the research by Jonah Mazet, Ian Gardner, David Jessup, and Linda Lowenstein on the effects of petroleum on mink populations to use as a model for reproductive success in sea otters for populations affected by oil spills. This research, published in 2001, was conducted jointly by the School of Veterinary Medicine at the University of California at Davis and the California Department of Fish and Game. It was published in the Journal of Wildlife Disease and has been cited by two other articles. The purpose of this research was to study the effect that petroleum products released from oil spills have on the reproductive success of female minks in a controlled environment. Two petroleum products commonly transported in the Prince William Sound region, Alaskan North Slope crude oil and Bunker C fuel oil, were used in the study to compare any differences in effects. They additionally tested the differences that acute dermal exposure to environmental oil versus chronic long-term ingestion through their food had on reproductive success. On March 24, 1989, the Exxon Valdez oil tanker struck a reef, damaging its hull and releasing 11 million gallons of crude oil into the Prince William Sound. This spill affected all animals in the oiled area, including sea otters, whose pup survival rate dropped during the following 4 years after the spill. Researchers from two different studies by Monnet and Roderman, published in 1992, and Belash et al., published in 1994, found that sea otter pup survival was lower for oiled areas than unoiled areas. Furthermore, the Belash et al. Study found that overall, pup survival for both oiled and unoiled areas had diminished from 1990 to 1991, and that the rate of survival increased from 1992 to 1993, showing that areas beyond those directly oiled had been impacted as well. However, in a separate study by Bodkin and Udovitz in 1992, researchers compared the ratios of adult sea otters to pups in both oiled and unoiled areas and concluded that oil exposure had no effect on reproduction. So, which finding is correct? With the results from these papers pointing to different findings, researchers wanted to acquire an accurate answer. One idea they had for explaining the results by Bodkin and Udovitz from 92 was that females from unoiled colonies could be reestablishing themselves in previously oiled sites, supplanting those that had died off from the exposure to the initial oil slick. Unfortunately, this hypothesis would be hard to prove without extensive tagging and tracking. Additionally, accurately assessing the effects of the petroleum products on a sufficient amount of wild sea otters in the field is extraordinarily difficult. In order to test their hypothesis, researchers set up a controlled experiment with a similar species to use in place of sea otters, mink, scientific name Mustela viscin. Mink raised on ranches were used as a substitute species for the experiment, as there are several similar characteristics between the two species. They are a member of the Mustelid family, are semi-aquatic, have extensive grooming behavior, a high metabolic rate, and are susceptible to numerous environmental contaminants. The petroleum types used in this experiment, Alaskan North Slope crude oil and Bunker C fuel oil, were selected as they are some of the most common fuels used and transported in the area. In this paper, Mazet et al. Hypothesized that only those female minks exposed through their food would simulate recolonization of previously oiled environments by sea otters. For the study, researchers randomly selected age-appropriate mink from a commercial breeding colony. The United States Department of Agriculture approved the housing, husbandry, and treatment used in the study. Animal health and welfare protocol for humane animal use was followed. All mink were housed in mesh cages that were connected to a nesting box. Mink were split into 5 groups: a control group exposed to seawater, a group dermally exposed to Alaskan North Slope crude oil, a group dermally exposed to Bunker C fuel oil, a group exposed to Alaskan North Slope crude oil through their diet, and a group exposed to Bunker C fuel oil through their diet. All mink were bred 60 days after the exposure event. Additionally, all males used for breeding had been proven to be successful breeders from past matings. Bi-monthly, researchers took blood, fur, and skin samples to test for quantities of total polycyclic aromatic hydrocarbons. For the dermal trial, 58 mink were tested. 10 were exposed to only seawater and used as a control group. 24 were dermally exposed to Alaskan North Slope crude oil and seawater. An additional 24 were exposed dermally to the Bunker C fuel oil. To emulate oil spill conditions, researchers exposed the mink to an oil-to-water mixture of 500 milliliters to every 4 liters of water. This created an oil slick that was 1.5 centimeters deep. The animals were exposed to the solution for 1 minute and allowed to move freely before being removed from the mixture. In order to simulate exposure from an oil spill event, oil was left intact on the dermis. Both experiment groups were exposed 60 days before breeding. This time delay allowed researchers to take bi-monthly samples of blood, fur, and skin for further testing. In total, only 33 of the 58 females from this group survived to breed. All 10 of the control group survived, 8 from the Alaskan North Slope crude oil group, and 15 from the Bunker C fuel oil group. Of those that did not survive, 11 died of acute effects of crude oil, 7 died of the acute effects of fuel oil, 5 from the crude oil group were euthanized for sampling protocols at weeks 1, 2, and 6, and lastly, 2 were euthanized from the fuel oil group for sampling protocols at weeks 1 and 6. After breeding occurred, females were not sampled any further. For the ingestion trial, 63 mink were tested. 15 were fed uncontaminated diets and were used as a control group. 24 were fed a diet that was contaminated with Alaskan North Slope crude oil, and 24 were fed a diet that was contaminated with Bunker C fuel oil. Per research conducted by Hartung in 1995, researchers contaminated the food with either crude oil or food oil at a concentration of 500 parts per million. Dietary exposure for both experimental groups began 60 days before breeding was commenced. Unlike the dermal trial, experiment groups were fed the contaminated diet through both breeding and pupping. Like the dermal trial, though, mink were subject to blood, fur, and skin sampling up until breeding commenced. In total, 59 survived to breed. All 15 of the control group, 22 of the crude oil group, and 22 of the fuel oil group. One of each experimental group died from exposure, and 1 from each group were euthanized for systematic sampling. At the end of week 6 for the crude oil group, and at the end of week 1 for the fuel oil group. Female mink were bred 60 days after either being exposed to oil spill conditions or after the start of the ingestion exposure diet. Breeding was conducted in accordance with standard mink husbandry practices. Females were paired with males for 2 consecutive days, and then again after a period of 1 week for an additional 2 consecutive days in order to maximize the potential for insemination. All males used for the experiment were previously proven to be successful breeders, removing any doubt from the possibility of for use of infertile males. After females started whelping, they were monitored by staff to accurately count the number of live births versus stillborn pups. As this was a blind study, the observers were not made aware of what sample group the birthing mother came from. Pups were additionally monitored for 21 days and until weaning to record kit survival. Overall, researchers compared the number of females bred, the gestation length, the number of females whelping, the number of live-born kits, the number of stillborn kits, the number of kits surviving to 21 days, and the number of kits surviving to weaning. Lastly, researchers examined histological sections for 6 females that bred and did not whelp, but were suspected to have conceived during the trial. The examinations were compared with 3 positive and 3 negative control specimens from the experiment. In addition to the original study, researchers randomly selected offspring from the surviving litters to record and compare their reproductive success. Overall, they used 83 female kits from the initial experiment to study, 40 offspring from the control group, 11 offspring from the dermal ANS crude oil females, 15 offspring from the dermal bunker C fuel oil females, 12 offspring of the ingestion ANS crude oil group, and 5 females of the ingestion bunker C fuel oil group. All females were randomly selected. In these groups, researchers compared the number of whelping females, number of live-born kits, number of stillborn kits, and the number of kits surviving to wean. Looking at Table 1, we see that the acute effects of dermal exposure caused a high death rate before the females even reached the breeding stage of the experiment. Excluding the 5 mink harvested to record organ damage in the ANS crude oil experimental group, 47.36% of the remaining mink died from exposure. Likewise, in the bunker C fuel oil group, excluding the 2 harvested for organ examination, 31.81% of the remaining mink died from initial exposure. Of those that bred, all females whelped kits. Additionally, the mean number of live-born kits per litter, kits surviving to 21 days, and kits surviving to weaning was not significantly different from the control group. Inverse to the dermal results, only 2 mink died from exposure to ingestion of the petroleum products, 1 from each experimental group. Whereas in the control and dermal females all successfully bred and whelped, the crude oil ingestion females had a reduced success rate for whelping. It should be noted, however, that the whelping difference was not significant from the dermal and control females. Bunker C fuel oil ingestion females, however, had significantly reduced success whelping. In both ingestion groups, the number of live-born kits per female was significantly fewer than the control and dermal groups. The difference was 57% less for ANS crude oil group, with a mean of 2.3 live-born kits per female, and 87% less for the bunker C fuel oil group, with a mean of 0.7 live-born kits per female. Kit survival to both 21 days and weaning in the crude oil exposure group was significantly lower than the both control group and the dermal bunker C fuel oil exposure group. In the bunker C exposure group, rate of kit survival once born was not significantly different from either the control group, the dermal groups, or the ingestion crude oil group. Reviewing Table 2, the offspring for both dermally exposed groups did not differ significantly from the offspring of the control group, with all dermal crude oil offspring successfully whelping with an average of 5.1 kits surviving to weaning. Although the dermal bunker C fuel oil group also did not differ significantly from the control group, only 14 of the 15 females successfully whelped, and average litter size surviving to weaning was nearly 1 fewer than the average for the dermal crude oil sample, and averaging 1.5 or 1.4 fewer kits than the control group. Reviewing the offspring for both ingestion groups, live-born litter size was significantly lower than the control group. Once born, though, the number of kits surviving to weaning was not significantly different from the other experimental and control groups. In the experiment, researchers only used petroleum concentrations of food at 500 parts per million, while levels recorded after the Exxon Valdez oil spill were measured at 1,250 parts per million in 1989 and were similar in 1990. Researchers wanted levels to reflect several years after an oil spill in order to observe long-term effects on populations. The average daily consumption of the contaminated food by the mink was 130 grams per day, which is 15 to 150 times lower than previous studies on the effects of petroleum products. When reviewing the results, one result that stuck out is the effect of ingesting bunker C fuel oil on the reproductive success of female mink. Overall, females were impregnated at a normal rate, but the pregnancies were more likely to self-terminate. They produced fewer kits per litter, and those kits that did survive to birth were less likely to survive to weaning than those from the control group. To confirm impregnation of the bunker C fuel oil females, researchers histologically examined 6 of the females that did not whelp and confirmed that 5 of the 6 had been impregnated during the experiment. The 6 showed signs of conception, such as fluid distended uterus and dark focal areas suggesting implantation sites, but these sites could not be confirmed. The impact of the effects of the bunker C fuel oil extends to the progeny of the ingestion experimental group. In the progeny study, females also had reduced number of kits per litter. This finding is interesting because the progeny had not been exposed to the fuel oil for over 9 months before reproducing, meaning that they were only exposed to the fuel oil transplacentally and through weaning. The effects on reproduction could be the result of either a reduced dose effect, reproductive impairment from development, or genotoxicity. These findings are important when considering recolonization of the areas exposed to oil spills and the generational effects on those not directly exposed to oil spills. The results of this paper reflect both the toxic acute effects and low-dose chronic effects of petroleum exposure in natural populations. Although dermal exposure to oil spill conditions lead to high mortality, the chronic effects on reproductive success were not found to be significantly different from the control groups. As this experiment was meant to establish a model for wild species, this research established that sea otter exposure to petroleum products reduced reproductive success through contamination of their diet. Lastly, sea otters recolonizing previously oiled areas will face reproductive issues from low-dose exposure to petroleum products still present in their food chain.